Greetings to all. In this lecture, we are going to see about cells of immune system, lymphoid lineage cells. Hematopoiesis is the process by which all cellular components of blood are formed and develop. This continuous process is essential for maintaining a steady supply of blood cells including red blood cells and white blood cells. All blood cells originate from a small population of hematopoietic stem cells. Hematopoietic stem cells are multipotent, that means they have the ability to differentiate into all various types of blood cells. They also have the capacity for self-renewal, ensuring a continuous supply of stem cells throughout an individual's life. The primary sites of blood cell production changes throughout this life. For example, during early embryonic development, it happens in yolk sac. During fetal development, it happens in liver and spleen. And during late fetal development and adulthood, it happens in bone marrow. By the end of the gestation, the bone marrow becomes the primary and lifelong site of hematopoiesis. In adult, active red bone marrow is mainly found in the axial skeleton and the proximal ends of the long bones, and sometimes extramedullary hematopoiesis also occur under certain pathological conditions. Hematopoiesis is a hierarchical process involving several stages of differentiation and maturation. Hematopoietic stem cells, the most primitive cells with self-renewal and multipotential, differentiate into multipotent progenitors, which have lost some self-renewal capacity but can still give rise to multiple lineages. They diverge into common myeloid progenitor and common lymphoid progenitor. Common myeloid progenitor and common lymphoid progenitor further diverge into lineage-specific progenitors. These progenitor mature into recognizable precursor cells. The precursor cells undergo further differentiation and it becomes the lymphoid lineage cells and the myeloid lineage cells. In lymphoid lineage cells, first we are going to see about natural killer cells. Natural killer cells are a type of cytotoxic lymphocyte and a crucial component of the innate immune system. Natural killer cells can recognize and kill certain target cells without prior exposure. This makes them essential for rapid responses against viral infections and cancerous cells. NK cells belong to the lymphocyte lineage, which also include T and B cells, but they develop and function differently. They are considered part of the innate immune system because they can recognize and respond to threats without prior antigen-specific recognition. Their primary mechanism of action is to directly kill target cells such as virus-infected cells and tumor cells. Morphologically, they are classified as large granular lymphocytes due to the presence of cytotoxic granules in their cytoplasm. Unlike T cells and B cells, NK cells do not express clonally rearranged antigen-specific receptors. Instead, they utilize a diverse array of activating and inhibitory receptors to recognize their targets. Natural killer cells employ a sophisticated system of activating and inhibitory receptors to determine whether a cell should be killed. This balance of signal is crucial for preventing NK cells from attacking healthy cells. Inhibitory receptors of NK cells. These receptors recognize major histocompatibility complex class 1 molecules present on the surface of the most healthy nucleated cells. MHC1 molecules present cell peptides signaling to the NK cells that the target is normal self cell and should not be killed. Many viruses and some tumor downregulate MHC1 expression to evade detection by the cytotoxic T lymphocytes. This downregulation makes them susceptible to natural killer cell killing because the inhibitory signal is lost. NK cells also have activating receptors. These receptors recognize various ligands expressed on stressed, infected, or cancerous cells. When activating signals outweigh inhibitory signals, the natural killer cell is triggered to kill the target cell. Mechanism of cell killing by the natural killer cell include release of cytotoxic granules. Natural killer cells contain specialized cytoplasmic granules filled with cytotoxic proteins. Upon activation, these granules are released by exocytosis and deliver their contents directly into the target cell surface. For example, perforin, a protein that inerts itself into the target cell membrane and forms pores, disrupting its integrity. And granzymes, a family of serine proteases that enter the target cells through the perforin pores and activate apoptotic pathway, leading to programmed cell death. The NK cell can also lead to induction of apoptosis via death receptors. Natural killer cells can also express death ligands on their surface. These ligands can bind to their corresponding death receptors on target cells, triggering intracellular signaling pathways that lead to apoptosis. Next, we are going to see about lymphocytes. Lymphocytes are a type of white blood cells crucial for body's immune system, produced in bone marrow and found in the blood and lymphatic system, including lymph nodes and spleen. They include B cells and T cells. T lymphocytes, also known as T cells, are a crucial type of white blood cells and central component of that active immune system. They play a vital role in recognizing specific foreign invaders and orchestrating a targeted immune response to eliminate them. The T cell stands for the thymus, the organ where these cells mature. T cell originated from hematopoietic stem cells in the bone marrow. Unlike B cells, which mature in the bone marrow, the T cells precursors migrate to the thymus for their maturation and education. In the thymus, these immature T cells undergo a rigorous selection process to ensure they can recognize self-MHC molecules and do not strongly react to self-antigens. T cells have antigen-specific receptors. Each mature T cells express a unique T cell receptors or TCR on its surface. This TCR can only recognize a specific peptide fragment of an antigen when it is presented by a major histocompatibility complex molecules on the surface of another cell. T cells cannot recognize soluble free antigens. T cells exhibit MHC restriction. T cells are MHC restricted, meaning their TCR can only bind to an antigen peptide that is bound to a specific type of MHC molecule. There are major subtypes of T lymphocytes: T helper cells, cytotoxic T cells, regulatory T cells, memory T cells. T helper cells or TH cells, they do not directly kill infected cells. They play a crucial regulatory role by releasing cytokines that help activate and direct other immune cells, including B cells, cytotoxic T cells, and macrophages. T helper cell differentiate into various effector subsets like Th1, Th2, Th17, THFH, and so on, each producing a different distinct set of cytokines and contributing to different types of immune response. Cytotoxic T cells. They are the primary effector cells of cell-mediated immunity, recognize and directly kill target cells that are infected with viruses or other intracellular pathogens, as well as cancer cell and transplanted cells. They kill by releasing cytotoxic granules containing protein like perforin and granzymes. Regulatory T cells. They play a crucial role in suppressing and regulating immune responses. They help maintain immune tolerance to self-antigens, preventing autoimmune diseases. They suppress the activity of other T cells, B cells, and antigen-presenting cells, helping to resolve inflammation once an infection is cleared. Memory T cells. They are all long-lived cells that develop after an initial encounter with an antigen, provide immunological memory, allowing for such a faster and more robust secondary immune response upon subsequent exposure to same antigen. They can rapidly differentiate into effector T cells upon re-stimulation. For the activation of T lymphocyte, the T cell requires two crucial signals from antigen presenting cells. Signal 1 is antigen recognition. The TCR on the T cell must bind to the specific antigen peptide presented by an MHC molecule on the antigen presenting cell. Signal 2, co-stimulatory signal. The T cell must receive additional co-stimulatory signals from the antigen presenting cells. Once activated, T cells undergo proliferation to generate a large population of antigen-specific effector and memory T cells and differentiation into their specialized subtypes. The next cell is B lymphocyte. The B lymphocyte, also known as B cells, are a crucial type of white blood cells and a central component of the adaptive immune system. They are primarily responsible for humoral immunity, which involves the production and secretion of antibodies. B cells are primarily responsible for humoral immunity, which involves the production and secretion of antibodies. These antibodies are specific to particular antigens and can neutralize pathogens, mark them for destruction by other immune cells, or activate the complement system. Some activated B cells differentiate into B memory cells, which provide long-lasting immunity by remembering previous encounters with specific antigen, allowing for a faster and more robust response upon re-exposure. B cell has got B cell receptor or BCR. Each B cell expresses thousands of identical BCR on its surface. The BCR is essentially a membrane-bound antibody molecule. It consists of immunoglobulin G molecule, the antigen binding portion responsible for recognizing and binding to a specific epitope or a part of an antigen. It also may have signal transduction molecules such as immunoglobulin alpha and immunoglobulin beta associated with immunoglobulin molecule. They transmit signals to the B cell upon antigen binding, initiating B cell activation. When a naive B cell encounters its specific antigen and receives the necessary activation signals, it differentiates into plasma cells. The plasma cells are highly specialized antibody secreting factories. Each plasma cell secretes large quantities of antibodies with the same antigen binding specificity as the original BCR on the naive B cell. Antibodies play a crucial role in eliminating pathogens involving neutralization, opsonization, complement activation, and antibody-dependent cell-mediated cytotoxicity. Following an initial immune response, some activated B cells differentiate into long-lived memory B cells. Memory B cells remember the specific antigen and can mount a much faster and more robust secondary immune response upon subsequent encounters with the same pathogen. In summary, B lymphocytes are essential cells of the adaptive immune system responsible for the humoral immunity. With this, we conclude the cells of the immune system lymphoid lineage. Thank you.